Today, folks, our main objectives are going to be able to talk about how the cell cycle is regulated. Okay? So our first objective is to be able to describe how the cell cycle is regulated. The second objective is to be able to briefly describe what's going on with cancer. I'm sure we've heard about cancer. Probably somebody has somebody in their family that's been affected by cancer. So we're going to discuss a little bit, just very briefly, of what is happening. Apoptosis. Be able to describe what that funky word means. Okay? And finally, we'll go into and be able to describe a stem cell and what stem cells do within our bodies. So let's start off here with the cell cycle regulation. The normal cell cycle. The normal cell cycle has proteins that are called cyclins that bind to enzymes called cyclin-dependent kinases during the start of interphase and mitosis. Those proteins help in the various activities of the cell cycle. So the cyclins bind to the cyclin-dependent kinases, which are enzymes. Everybody remember what an enzyme is? It helps to speed up a reaction. We said an enzyme is a biological catalyst. It speeds up the reaction. Remember when I showed you at the back of the room when I had the hydrogen peroxide and I added I forget what it was, something iodide, sodium iodide maybe, and it shot out of the test tube or the graduated cylinder. Okay? Forget what that was. I'll have to look that up. But I think it might have been sodium iodide. I still got it on my cart back there. So I'll take a peek here in a second. But nonetheless, enzymes are biological catalysts. They help to speed up a reaction. We have what we call checkpoints. Okay, it's listed right there, quality control checkpoints. I was right, I just looked. That is sodium iodide I used. Quality control checkpoints. They monitor the cycle and can stop it, meaning the cycle, if something goes wrong. So we have these checkpoints on the cell cycle, and if something bad happens, if something goes wrong, these checkpoints will prevent the cell cycle from going any further. Okay? If you look at this, what I have right up here, if you look at this, these things should look familiar, and you're going to put this diagram into your interactive notebook tomorrow. So we're going to cut, color, and paste this diagram. But the things look familiar here. Here's G1. Here's S phase, the synthesis. Here's G2. What do you think this M stands for? What does the M stand for? What have we been talking about here? Mitosis, okay? So G1S and G2 are all interphase, and M is mitosis. Now, what this looks like to me, And I don't know about you folks, do you guys ever do laundry? Anybody here do their own laundry? Doesn't this kind of look like the control on your laundry? Okay, kind of? Can you see a little bit? Okay, so it's going on, and we're going through our control system, and we're getting at certain points. Let's start right here. Okay, the cell just divided. We're in interphase. We're chilling out. We're going around. All of a sudden, bam, we hit the first checkpoint. Okay, if something went wrong on this checkpoint right here, the cell cycle would stop for that particular cell. Okay, so each individual cell are going to have these checkpoints. So that checkpoint right there, bam, we hit it. It stops. Something's wrong, it stops. Okay, we won't get into what happens then. Okay, it goes into what we call a G0 phase. And it just pumps off, it stops there, and it will never reproduce a cell again. Okay, or it can go dormant for amounts of times. Okay, let's say it passes this checkpoint. We got the green light here, the go. So we get the go at the G1 checkpoint. We go through the S phase, which is also the synthesis phase. So the DNA is replicated. The chromosomes are replicated. We go into the G2 phase, and all of a sudden, bam, here's the G2 checkpoint. So we have a checkpoint and say, okay, is everything ready to go for us to start mitosis, which is the division of the genetic material and cytokinesis? Is everything okay to go here? If we get the green light, then we go through the phases of mitosis. Remember, how do we remember the phases of mitosis? PMAT. Okay, prophase, metaphase, anaphase, telophase, and then we go into cytokinesis. After that, then we have an M checkpoint right here. Okay, before we start the division, before we start cytokinesis, we have this M checkpoint. Is everything good to go? And then we start the cycle again. Okay, so these checkpoints are what helps the cell to regulate itself. Now let's talk about normal cells versus cancer cells. A normal cell versus a cancer cell. Well, and that's kind of tough to read, but that says abnormal cell cycle. It's in your notes there, right? You'll have to write that down. Abnormal cell cycle is what we call cancer. Cancer is the uncontrolled growth and division of cells. Uncontrolled growth and division of cells. It can kill an organism by crowding out normal cells, resulting in the loss of tissue function. Can crowd out the normal cells, causing a loss in the function of the tissue. Causes of cancer are things that we call mutations, and we can also have environmental factors that cause cancer. I'm sure we can think of an environmental factor that causes cancer, can't we? Anybody? Huh? Yeah, smoking. Okay. Smoking is an environmental factor that could cause cancer. Okay. Have you ever heard of skin cancer? What causes that? Huh? Being exposed to sun and the ultraviolet lights. Okay. So we're going to talk about some of these mutations and these environmental factors. All right. But first, let's take a look at these two cells. Okay, or actually these are 3 cells, but if you look here, here are the normal cells. Okay, you can see that we got nucleus, we got chromatin in there, we got the cell membranes and stuff, cytoplasms. Okay, look how large, how large the nuclei are, in this one. And look at this one. This particular cell has what? It has 2 nuclei, doesn't it? So something happened in division there in the cytokinesis that sort of got messed up. Okay? So the cytoplasms are much smaller here in the cancer cells. Okay? They have multiple nuclei. Okay? And there's difference between the chromatin in the nuclei. Now, if we look here, here I have lists of some different carcinogens. And what that is, a carcinogen is just a cancer-causing agent. Okay? Pesticides, aerosols. We talked about ultraviolet rays, radiation, okay? Vehicular emissions, okay? Talk about smog out on the West Coast, out by LA. That comes from vehicular emissions. How about some foods? Okay? Some foods, different preservatives. Okay? Sweetened juices, some sweeteners could be carcinogens. Okay? Dietary fat, highly spicy foods, fermented foods, some lifestyle things here. Chewing tobacco, smoking, okay? Industry, paints, okay? You ever heard of asbestos? You ever heard of that? We don't have much asbestos around anymore, but it used to be, I remember at one time that part of ceilings in schools were made of asbestos. So they had to tear it all out. Okay? Asbestos, I think, were used some in brakes, also some in installation, or not installation, insulation. Okay, around pipes, the likes of that. So asbestos. Okay? Gamma radiation, viruses, okay? Herpes, hepatitis B and C, okay? The HPV virus. So these are all different things that could potentially cause cancer, okay? Here we go. I put this in here. Oh, I got to turn up the volume, okay? I got to turn up the volume on my radio. I forgot to do that. Okay, here we go with apoptosis. Okay, what is apoptosis? It is the programmed death of cells. Do you think that cells actually die? Well, they do. Okay? Stop and think about, if you scratch, aren't you scratching off skin cells? Okay, you can scratch skin cells. You can scratch skin cells off your body. Those cells are no longer working, okay? Cells will shrink and shrivel in a controlled process that we call apoptosis. All cells have this death program that can be activated. part of the developmental cycle of the cells, and it can protect the organism by killing cancer growths. So let's say that, okay, one of these cells has an abnormality. Okay? Has an abnormality. We talked about the cellular regulations, right? The checkpoints. Okay? Well, maybe at a checkpoint that the cell detects that there's an irregularity and it starts into this apoptosis, the programmed death. Not a big deal. We got something wrong with the cell goes into the programmed death, and we never see any problem with it. Okay? But the problem could be, what if it doesn't? What if it doesn't go through that programmed death? That's where we're going to see the abnormal cells start reproducing, and all of a sudden, all of these cells that are abnormal reproducing turn into a growth. Okay? Turn into a growth. And maybe, it could be a lump on someone's breast with breast cancer. Could be a lump in somebody's lung. Okay? Those abnormal cells that keep reproducing and don't have this programmed death. Okay? Could it be then? Could it be that the abnormal cell spread to other parts of the body? Yeah. Yeah, because cancer could spread. Somebody could have lung cancer and then it spreads into the stomach and have stomach cancer. And it could spread all over the body. Okay? Those abnormal cells get into the blood system, okay, and start going to different places of the body. Okay? So this apoptosis, I call it apoptosis, okay, is the programmed death that can help prevent that. Okay? Can't see that very good, but you see here is a normal cell. Here is the cell that is condensing, then it is fragmenting, and then actually, folks, some of those parts of the cell, it's kind of like a chop shop with a car, okay? Or a junkyard with a car. Could you take something off a car that's a piece of junk that might work on a different car? Yeah. Well, what happens when the cell is going through this programmed death? Some parts of the cell can be recycled and reused. Okay? Some different organelle could possibly be reused in a different cell. Finally, we're going to talk about stem cells. Okay? We're going to talk about stem cells. Stem cells are unspecialized cells that can develop into specialized cells when under the right conditions. So they're unspecialized cells that can develop into specialized cells. What would be a specialized cell? Maybe a blood cell. Okay, maybe a nerve cell, maybe a muscle cell. Okay, those are some specialized types of cells, but these stem cells are unspecialized. So if I look here, I have the stem cells right here. Well, maybe they turn into intestinal cells, liver, okay, cardiac cells, nerve cells, blood cells, muscle cells. So those are all on the outside here, the specialized cells that when these stem cells are in the right condition, they might change into the specialized types of cells. Embryonic stem cells. After fertilization, there's a resulting mass of cells that divide repeatedly until there's about 100 to 150 cells. These cells are not yet specialized, and they are easy to manipulate into desired cells. Embryonic stem cells. Now, if you look right here, the picture I have right here is an in vitro egg that was fertilized. What in vitro means, it was fertilized like in a test tube. Okay? So a scientist has combined a sperm and an egg, and here is the embryo that was formed. This mass of cells right here are the embryonic stem cells. Okay? That they can be, they're unspecialized cells, but they can be, like it says, easily manipulated into other types of cells. Okay? There's a lot of controversy with this embryonic stem cell stuff. Okay? We're not going to get into that. I just wanted you to know what they are. Okay? What they are. Adult stem cells. Okay? Adult stem cells, they are found in various tissues in the body. And they may be used to maintain and repair the same kind of tissues. So these adult cells are in our body, the adult stem cells, and they may be used to maintain and repair. They are a lot less controversial than embryonic stem cells. However, they are a lot more difficult to manipulate. They're not as easily changed into the specialized types of cells. Okay? If you look here, okay? If we look here, here is looks like a hip. Here's a hip socket, okay? Here is the upper leg bone. What is that? The fibula? No. Fibula, the femur. The femur. Went into a little brain shock there. The femur right here. They can take bone marrow, and in that bone marrow of the femur, we have stem cells. And they could possibly specialize those adult stem cells into liver cells, heart cells, muscle cells, brain cells, nerve cells. But like I said, it's a lot tougher to manipulate those cells than it is the embryonic stem cells. Okay? Now, that